clicked on this because you are tired of being told to eat less. You have restricted. You have cut carbs. You have done the cardio, and the belly fat is still there. Here is what no one is telling you. Restricting calories is not just ineffective for belly fat. In many cases, it is the reason it will not leave. The last 5 years of research have completely changed how we understand where belly fat comes from, why it stays, and what actually shrinks it. And almost none of it has to do with eating less. This is not a motivational video. This is not a just believe in yourself video. This is the science, the actual studies, and what they say you should do instead. Because the old model was wrong. And the sooner you understand why, the sooner your body can actually change. Let's start with the thing everyone gets backwards. Most people think belly fat is simply stored energy. You eat too much, the excess gets packed away around your abdomen, and you burn it off by eating less. That model is clean. It is logical. It is also dangerously incomplete. Because belly fat, specifically the fat that sits deep inside your abdominal cavity, wrapping around your organs, is not passive storage. It is a living, metabolically active organ. And the research from the last 5 years has made that clearer than ever. In 2022, a landmark paper published in Nature Metabolism identified something researchers had suspected but never fully confirmed. Visceral fat, the deep belly fat, operates almost like an independent endocrine gland. It secretes hormones. It releases inflammatory signals called cytokines. It communicates directly with your brain, your liver, your pancreas. It is not waiting for you to burn it. It is actively defending itself. And one of the primary tools it uses to defend itself is cortisol. Here is where the restriction story starts to fall apart. When you cut calories aggressively, say below 1,200 calories per day, which is what most popular diets recommend, your body registers that as a survival threat, not a health choice. A threat. Your hypothalamus triggers a stress cascade. Cortisol rises, and cortisol, particularly chronic cortisol elevation, is one of the most well-documented drivers of visceral fat accumulation in the entire scientific literature. A 2023 meta-analysis in Obesity Reviews looked at 41 studies on caloric restriction and cortisol. The finding was unambiguous. Severe caloric restriction reliably elevates cortisol in healthy adults, and that elevation is proportional to the degree of restriction. The more aggressive the cut, the higher the cortisol. And higher cortisol means more belly fat, not less. So when you go on a hard diet and you feel like your belly is not responding, or worse, it seems to be getting a little thicker, even as the rest of you shrinks, that is not your imagination. That is cortisol doing exactly what the research predicts it will do. But cortisol is only part of the story. Because in the last 5 years, researchers have uncovered a second major driver of visceral fat that almost nobody is talking about outside of academic circles. And it has to do with something happening not in your fat cells, but in your gut. The microbiome research of the last decade set the stage. But it was a series of studies between 2021 and 2024, that really tightened the connection between specific gut bacteria and belly fat accumulation. The short version is this. Certain bacterial populations in your gut, particularly from the Bacteroidetes phylum, produce short-chain fatty acids like butyrate that directly influence how your body partitions fat. When these populations are healthy, butyrate signals the liver to oxidize fat rather than store it. When they are depleted, which happens with ultra-processed food, chronic stress, antibiotics, poor sleep, the signaling breaks down and fat storage, especially visceral fat storage, increases. A 2023 study out of King's College London followed over 1,000 adult twins and found that the diversity of the gut microbiome was a stronger predictor of visceral fat accumulation than either total caloric intake or physical activity level. Read that again. Your gut bacteria composition predicted belly fat better than how much you ate or how much you exercised. That does not mean calories and exercise are irrelevant. It means they are not the whole story, and possibly not even the most important chapter. This matters because of what kills microbiome diversity fastest. It is not eating a little too much. It is 3 things: ultra-processed food, chronic sleep deprivation, and, here is the irony, extreme caloric restriction. When you severely restrict food intake, you reduce the variety of substrates your gut bacteria have to feed on. Diversity collapses. The butyrate producing strains decline, and the conditions for visceral fat accumulation improve. Once again, the restriction strategy is working against the very outcome you want. Now, let's talk about insulin. Because this is where the last 5 years of research have produced what might be the single biggest shift in how scientists understand belly fat. For a long time, the conversation around insulin and fat was framed around insulin resistance as a consequence of obesity. You get fat, you become insulin resistant, then your fat accumulation gets worse. That was the sequence most researchers accepted. But newer mechanistic research, particularly a series of studies using hyperinsulinemic euglycemic clamp techniques, which are the gold standard for measuring insulin sensitivity, has started to flip that sequence around. The emerging picture is that insulin dysregulation, specifically what researchers now call postprandial hyperinsulinemia, may precede and cause the belly fat accumulation, not the other way around. What is postprandial hyperinsulinemia? It means that in response to eating, your body releases more insulin than the meal actually requires. Your blood glucose comes back down to normal, so you would test as fine on a standard fasting glucose test. But in the hours after eating, your insulin has spiked well above what is needed, and that excess insulin is doing what insulin always does. It is telling your fat cells, including your visceral fat cells, to store. A 2022 study in Cell Metabolism followed healthy, non-obese adults over 18 months. The participants who showed high postprandial insulin responses to standardized meals at the beginning of the study gained significantly more visceral fat over the study period than those with lower responses, even when their total caloric intake was identical. Same calories, more belly fat, driven by insulin dynamics alone. The practical implication of this is significant, and it is one that very few mainstream health voices have caught up with yet. It is not just about how much you eat. It is about the hormonal signal your food produces. 2 meals with identical calories can produce radically different insulin curves depending on their composition, timing, and even the order in which you eat the food on your plate. A phenomenon called food sequencing, which has its own growing body of research we will get to in a moment. Before that, let's address the exercise question, because the last 5 years have added real nuance here that changes the recommendation in a meaningful way. The old advice was simple. Do cardio to burn fat, and cardio does burn calories during the session. But a 2021 study in the Journal of Hepatology, focused on visceral fat specifically, compared 3 groups. One doing steady-state cardio, one doing resistance training, and one doing a combination. After 16 weeks, the cardio group had lost the most total weight, but the resistance training group and the combination group had lost significantly more visceral fat, even in cases where total weight loss was lower. The reason comes down to something called myokines. When your muscle contracts under load, meaning actual resistance, actual effort, they release signaling molecules called myokines. One of them, interleukin-6, released in the acute context of exercise rather than chronic inflammation, directly acts on visceral fat cells to promote lipolysis, which is the breakdown of stored fat. Another myokine, irisin, crosses the blood-brain barrier and has been shown to improve insulin sensitivity, specifically in visceral fat tissue. Cardio does not produce myokines at anywhere near the same level. Walking is wonderful for your health in many ways, but it is not producing the muscular contraction intensity needed to generate a meaningful myokine response. This is why the studies keep showing that people who prioritize muscle, building it, preserving it, tend to have less visceral fat at equivalent body weights than people who prioritize cardio. But here is the debate that is still very much alive in the literature. Some researchers argue that the key variable is not the type of exercise at all, but the total weekly energy expenditure. If you do enough cardio, they argue, you will produce comparable outcomes to resistance training for visceral fat. The counter-argument, and this is where it gets genuinely contested, is that resistance training changes the resting metabolic environment in ways that cardio does not, particularly in how skeletal muscle handles glucose disposal. More muscle means more tissue that can take glucose out of the bloodstream without requiring insulin to do it, which directly reduces the postprandial insulin spikes we discussed earlier. The debate is not settled, but the preponderance of recent evidence leans toward resistance training as the more targeted tool for visceral fat specifically, while cardio remains valuable for cardiovascular health and overall energy balance. Now, the food sequencing research, because this has been one of the most practical and underreported findings in nutrition science over the last few years. Researchers at Cornell and independently in Japan have been studying what happens when you change the order in which you eat the components of a meal. Not the meal itself, not the calories, just the sequence. The findings are striking. When people eat the protein and fat and fiber-rich vegetables of a meal first, and the carbohydrates last, the postprandial glucose response is blunted by as much as 30 to 40% compared to eating carbohydrates first. The insulin response follows the same pattern, same meal, same calories. Dramatically different hormonal outcome, simply by starting with the steak or the eggs or the salad before touching the bread or the rice. The mechanism is reasonably well understood. Protein consumed first triggers the release of glucagon-like peptide, 1 and peptide, Y, from the gut, which slows gastric emptying and signals the pancreas to moderate its insulin response. Fat and fiber consumed early in the meal physically slow the rate at which glucose enters the bloodstream. By the time the carbohydrates arrive, the machinery for handling them is already primed and calibrated, rather than overwhelmed. This is not a radical dietary overhaul. It does not require counting anything. It is a sequencing change, and the evidence for its impact on the hormonal environment, and specifically on visceral fat over time, is accumulating quickly. Let's talk about sleep. Because a 2023 paper in Nature Communications did something that should have been front-page news and was barely covered. Researchers took healthy, lean adults with no metabolic problems and had them sleep for 6 hours a night for 2 weeks, compared to a control group sleeping 8 hours. Then they used imaging to measure visceral fat directly. The sleep-restricted group accumulated measurably more visceral fat over those 2 weeks. Not subcutaneous fat, not overall weight, specifically visceral fat. And the primary mechanism identified was, again, cortisol, combined with elevated ghrelin and suppressed leptin, which together produce stronger cravings for high-sugar, high-fat foods, even when total caloric intake was controlled. Sleep deprivation does not just make you hungrier. It specifically drives the hormonal conditions that tell your body to store fat in the abdomen. The research also found something interesting about the timing of fat accumulation under sleep restriction. Visceral fat increased fastest in the first 4 to 5 days of restriction, then plateaued somewhat. This suggests the body adapts, not in a good way, but in a way that stabilizes the cortisol response at an elevated level, which means the damage is not primarily accumulative in a simple linear sense. And it is front-loaded. 1 week of consistently poor sleep can shift your visceral fat dynamics in ways that take considerably longer to reverse. Now, let's bring this all together, because the picture that emerges from 5 years of research is coherent, and it is different from what most people have been told. Belly fat, specifically visceral fat, is not primarily a calorie problem. It is a hormonal environment problem. The hormonal environment is shaped by cortisol, by insulin dynamics, by myokine signaling from muscle, by the gut microbiome, and by sleep. And the critical insight is that the traditional intervention, eat less and do cardio, directly worsens at least 3 of those 5 factors. It raises cortisol through restriction stress. It fails to generate the myokine signaling that targets visceral fat specifically. And because it is often paired with poor sleep due to hunger and metabolic disruption, it compounds the visceral fat-promoting hormonal environment rather than correcting it. What does the research actually point toward? A few consistent themes emerge across multiple studies and multiple research teams. First, protein adequacy over caloric restriction. The studies consistently show that increasing protein to at least 1.6 to 2 grams per kilogram of body weight, without necessarily reducing calories, improves body composition, specifically visceral fat, better than caloric restriction with lower protein. The mechanism is partly satiety, partly the thermic effect of food, partly improved insulin sensitivity, and partly the preservation and growth of muscle tissue. Second, resistance training as the primary exercise modality for visceral fat. Not as an add-on, but as the anchor. 3 to 4 sessions per week of progressive resistance training appears in the literature as the most consistent, non-pharmacological intervention for reducing visceral fat across all age groups studied. Third, sleep treated as a biological priority, not a lifestyle preference. The research is unambiguous that chronic sleep below 7 hours elevates visceral fat-promoting hormones. This is not a secondary variable. In some studies, correcting sleep alone produced measurable reductions in visceral fat without any dietary intervention. Fourth, and this is where the microbiome research lands practically, increasing dietary diversity, particularly of fermented foods and prebiotic fibers, to support the bacterial populations that produce butyrate and regulate fat partitioning. This does not require supplements. Consistent intake of foods like kefir, kimchi, sauerkraut, and a wide variety of plant fibers has been shown in multiple randomized controlled trials to measurably improve gut microbiome diversity within 4 to 6 weeks. And that shift in microbiome diversity correlates in the literature with reductions in visceral fat accumulation rate. Fifth, managing the postprandial insulin environment through food sequencing and the strategic use of lower glycemic carbohydrate sources. Not eliminating carbohydrates, but timing and sequencing them to reduce the hormonal overshoot that drives visceral fat storage. Here is the hardest truth of everything covered in this video, and it is the one that the research keeps circling back to in different ways. Belly fat does not respond well to urgency. The hormonal systems involved, cortisol in particular, are exquisitely sensitive to stress, and aggressive fat loss attempts are metabolically stressful. The studies on slow, sustainable approaches versus rapid restriction consistently show that rapid restriction produces faster initial results and worse long-term outcomes, particularly for visceral fat. Because the cortisol elevation that comes with aggressive restriction actively opposes visceral fat loss, even as it may produce short-term reductions in subcutaneous fat. The body is not being irrational. It is doing exactly what millions of years of evolution programmed it to do. When resources are scarce, protect the vital organs. And the fat that wraps around your organs is, from your body's evolutionary perspective, an emergency reserve. Stress hormones tell your body to keep that reserve intact, which means the counterintuitive truth that the research keeps arriving at is this: the path to less belly fat is not more restriction, more stress, more urgency. It is a more supportive metabolic environment. More muscle, more sleep, more microbiome diversity, better hormonal signaling, adequate protein, and an insulin environment that is not chronically spiked by refined carbohydrates, eaten in ways that overwhelm the system. None of that is a magic solution. None of it works in a week, but unlike the restriction model, it works with your physiology rather than against it, and the studies, accumulated over the last 5 years, are increasingly clear on which approach actually shrinks visceral fat and keeps it off. The answer is not to eat less and suffer more. The answer is to build a body that is hormonally, metabolically, and biologically incompatible with holding on to that fat. That is a different project. And it starts with understanding what you just heard.